red, and red is going to represent the lipids. So anything that's a lipid is going to be in red. Anything that is a protein is going to be in blue. And anything that is a carbohydrate is going to be in green. Okay, so up until now, we have interchangeably used, hopefully I haven't, but I've allowed you to, use the term phospholipid bilayer and plasma membrane, okay? Those terms are no longer going to be interchangeable because they are not interchangeable. They're very different things, and hopefully after today you will understand exactly why that's the case. These guys here that have a phosphate head and they have fatty acid tails, those things are the phospholipids, and they are in a bilayer. But the plasma membrane doesn't just include those things, it includes a lot of other things too. So we're going to draw some other stuff in here. Here's a big protein here, and this protein is going to travel through the whole membrane. It's going to provide a little hole for stuff to travel through. A channel, right? Then we got some more phospholipids, like this. Maybe in the middle of these phospholipids there's going to be some big structure in here that looks something like this. It's also red. It's a lipid. And there's some phospholipids around it like this. Then maybe we've got a protein that's in here like this. Just embedded in one side. Maybe another one of those big ugly lipids. This phospholipids going all around it. Maybe over here there is another protein, but this protein is just kind of like hanging out outside of the membrane like this. We got more phospholipids in here. Something like this. And the last thing we see, we haven't used any of our green yet. The green is actually going to be on the outside, embedded into different parts like this. Some of them are attached to proteins. Some of them are attached to phospholipids. There's no rhyme or reason to how many little branches I'm putting on these things. Something like this. And this represents our plasma membrane. Okay, the plasma membrane, as you can see, consists of 3 large molecule types. Those are lipids, proteins, and carbohydrates. So let's say the plasma membrane made from lipids, proteins, and carbohydrates. And then I'm going to take a drink here and allow you guys to finish drawing and writing and stuff. I hear a lot of clicking going on still. Alright, so the first group that we're going to talk about are the lipids. And the first lipid that we drew on there was the one that looked like this. This lipid is called a phospholipid. We've talked about it in a fair amount of detail already. We know that phospholipids are a phosphate head and 2 fatty acid tails. And they make up the phospholipid bilayer. And the phospholipid bilayer is extremely important because that's going to be the thing that acts as a barrier between the outside of the cell and the inside of the cell. Acts as a barrier between the outside and inside of the cell. And you may have read that the phospholipid bilayer, or maybe even the plasma membrane, is semi-permeable, meaning it's going to allow some stuff in and some stuff not to come in. But the only thing that the phospholipid bilayer allows through are things that are small and nonpolar. So only small and nonpolar molecules. Can diffuse through. And there are actually not that many molecules that fall under these two categories of small and nonpolar. But there are 2 of them that are extremely important that turns out for life is really important that they can go through. And those are carbon dioxide, which is small and nonpolar, and oxygen, diatomic oxygen. Okay, and we know that diatomic oxygen is needed for cellular respiration, and carbon dioxide is a waste product of cellular respiration. So it's good that it can freely diffuse in and out. Right? And we know that it's the opposite for photosynthesis, that they're going to need carbon dioxide and they're going to produce oxygen as a byproduct or as a waste product. And so it's, again, good that those can diffuse in and out freely because you don't want your waste product to build up, and you need to have that starting product, or that starting reactant, rather, to allow for the reaction to happen in the first place. Okay? We know that in order for the phospholipid bilayer to work properly, it needs to maintain the optimal fluidity, or proper fluidity. Say it must maintain optimal fluidity. And we've already talked about the way that happens by changing the ratio of saturated fatty acid tails to unsaturated fatty acid tails. It's going to be what mostly will control the fluidity. Saturated, unsaturated fatty acid tails. And when I say this, when I say optimal fluidity, I want to talk about what that means for a second here. Okay? So up here in our drawing, this thing is sometimes called, or sometimes referred to as the fluid mosaic model. And what that means is that each of the layers of this phospholipid bilayer is going to behave almost like a fluid. It's going to be able to actually move around within this section. So if we looked at one specific, you don't have to highlight here, but if we looked at this phospholipid here, this phospholipid here is not likely to be right there in 5 seconds, right? 5 seconds later, it might be all the way over here, right? Or then over here, and it's going to be flowing around constantly. What it's not likely to do is it's not likely to flip to the other side. And the reason for that is, in order to get to the other side, this phospholipid, or this phosphate head rather, would have to travel through this fatty acid tail middle part here to pop out on the other side. And so it's very unlikely that it will do that. It's much more likely that it will just move around in that area on the top. This is what we mean when we're talking about fluidity. So we want them to move around, but we don't want them to move around too much, right? All right. So the next thing is going to help us with that, and the next thing that we drew sort of looked like a little like crooked box of some kind like that. This is another type of lipid that we learned as a steroid, and it's a specific type of steroid that's called cholesterol, which is a steroid. And the main job of cholesterol is to control the fluidity, make it so it's not too fluid and make it so it's not too solid. The way that I always refer to this as is like a buffer. Okay, so a buffer is going to be anything that resists change, and so we talk about like pH buffers and stuff like that, but this is going to be like a fluidity buffer. So we can say that it buffers the fluidity of the membrane. And so it's going to be able to act on it in two ways. The first way is if the membrane is too fluid. So if the membrane is too fluid, the cholesterol blocks the movement of the fatty acid tails. So I would equate this to like a breaker or something on a beach. So like a lot of beaches have really rough seas around them, and so in order to make the beach not erode, in order to make it better for people to actually go to the beach, you wouldn't want to have these massive crashing waves. And so a lot of times they'll go out and they'll build like a little sandbar, or they'll build like a little wall out there, and what that does is it causes the waves to break on the sandbar first, so that on the other side of it, it's not as wavy. It's nice and calm, right? And the same thing goes here for this cholesterol. So if you can imagine, this is like your breaker, your wall. And as these fatty acid tails are moving around, every time they have to go past the cholesterol molecule, they crash into it, and that's going to slow them down so that they're not moving around as fast, right? And that's going to reduce the fluidity of the membrane if it ever starts going too fast. Same thing can happen, or opposite thing rather, can happen if it's going to, or if they're becoming too solid. Say if the membrane is too dense, it prevents the fatty acid tails from clumping together. Okay, and so that's going to stop it from getting too dense or too close together. Yeah.
So why is high cholesterol bad?
It's not bad for you at all. There are certain types of cholesterol that when they get processed by your body can cause deposits of what we call plaque in your arteries, and so can cause things like, yeah, like arterial blockage, which can lead to heart disease. But we're kind of maybe thinking that that's not true, that it might not be diet related at all. Certain people produce more cholesterol than others, and it's blood-borne cholesterol versus any other type of cholesterol. A lot of times when you eat cholesterol, let's say I go out and I eat 60 eggs or something like that, that's going to be a ton of cholesterol intake in my body. Certain people will see a small rise in their blood cholesterol level, whereas other people will see no rise in blood cholesterol. But if you just took their resting blood cholesterol in those two people, one of them would be higher anyways, even without eating the high cholesterol diet. So the link between dietary intake of cholesterol and heart disease is not 100% proven at this point. In fact, it's moving towards the other way, where it's becoming more and more disproven. Yeah.
I was going to say, so there's like no correlation between the two?
Yes, we're finding less and less correlation between the two. All right, but cholesterol is super important. Like, you absolutely need cholesterol. Every single cell in your body has cholesterol. All of your Sex hormones use cholesterol as their building block, so like you need it. Yeah.
So what does the medicine do, like to lower your cholesterol?
The medicines are mostly blood thinners, which means that even though the cholesterol's in your blood, it is allowing your blood to sort of travel a little bit faster, so that there's not as much deposits, so it doesn't actually like keep building up on the walls. Yeah. All right. So next, proteins. So there's a couple different types of proteins, and we've drawn a couple different types. The first type we're going to talk about are integral proteins. Integral proteins are any protein that is embedded in the membrane. In the plasma membrane. Okay, so we've drawn two that were embedded. One of them was just an integral protein. So the one that looked like this, that had the flat top, right, that was an integral protein. Okay, and that is different than the other one that we drew that was an integral protein that has another classification towards it, and that is called a transmembrane protein. And transmembrane proteins are ones that travel through both sides of the bilayer. So the one that was like that was the channel protein that we drew. Sort of like a hot dog bun. Okay, and a transmembrane protein, this is specifically a transmembrane channel protein. And a transmembrane channel protein is going to allow for the diffusion of things that would normally not be able to diffuse through the membrane. So allows for molecules that are not small and nonpolar to diffuse through the membrane. And there's one specific small, there's one molecule that is small but very polar that's absolutely required for all cells, and that is water, right? Cells need to be able to transport water in and out, and so in order to do that, there has to be a channel protein that is specific for water, and every single cell has that channel protein. Now, they're in varying numbers in some cells, but every single cell has them, and they're called aquaporins. And aquaporins are specific channel proteins that are for the transport of water. Allows for passive transport of water. So this passive transport is important because that means that no energy is required. It's going to use diffusion, specifically a type of diffusion called facilitated diffusion, we will talk about tomorrow. Okay. There's another channel protein we've talked about in here. Anybody know the other channel protein that we've talked about in fairly long length in here? Came up a bunch, especially when we were talking about chemiosmosis. ATP synthase is technically a channel protein. Right? When we talked about the power-producing organelles, right? We said that there was these black proteins that were embedded here, right, in the thylakoids. And these proteins were going to allow H plus ions to travel out of the membrane, even though H plus ions are not small and nonpolar. They're very polar. They're ions, right? And so they have to go through something. Specifically, they're going to go through ATP synthase, right? So it allows for the transport of H plus ions. Right? And then you got your other class of proteins, which are called peripheral proteins. And peripheral proteins are non-embedded. Non-embedded proteins that can travel through the membrane. We're going to talk about two varieties of them. One of them is called a signaling protein. Okay? Signaling proteins are going to carry non-permeable cell signals from the outside of the cell into the cell. From the outside to the inside of the cell. And the way that they do that is by just binding to them and then freely traveling throughout that plasma membrane. So if we go back up here, imagine there's some cell signal. Here's some cell signal, right? And this cell signal is trying to travel through the membrane, but it can't because it's not small and nonpolar. It could be picked up by this guy here, right? It could go into that one, and that protein could then travel through the membrane like that, right? And that would be an example of a signaling protein because this protein has then moved through the membrane and brought the signal to the other side, okay? It's just our little circular protein, okay? And the other type of this is a permease, which is a type of... there we go. Finish tomorrow. Are sometimes called carrier proteins. And permeases are molecules that are going to assist in cell transport or transport into the cell or out of the cell of molecules. And they're going to do that by wrapping around those molecules and making them permeable so that they can travel through the membrane. Okay, so these are proteins that come on, computer. Okay, proteins that move non-permeable molecules into or out of the cell by wrapping around them and traveling through the membrane. Okay, so for instance, if you had like some molecule up here, I think I already drew it. I don't remember doing that, but some molecule that it would normally be impermeable that can't go through the membrane, it can attach to this carrier protein, this permease here, and the permease itself can attach or can move through the plasma membrane and then deposit that molecule on the inside of the on the inside of the cell. Okay, it's a type of diffusion called facilitated diffusion. All right, so let's talk about the last category then, which are the carbohydrates. You might see these carbohydrates called oligosaccharides. The term saccharide is one that you're familiar with. It just means sugar, right? And then the prefix there, oligo, means short. So these are short sugar chains. And they come in two different types. First type is called a glycoprotein. Okay, and this is a little confusing because a glycoprotein sounds like it should be a protein, but it's not. It's a carbohydrate. Specifically, this is a carbohydrate that is attached to a protein. So a short sugar chain attached.
Can you pronounce that word again?
Glycoprotein.
No.
Oligosaccharide.
Okay.
To a protein. And then the other type is called a glycolipid. And that is a short sugar chain attached to a lipid. Now, for the purposes of this class, it is not important for you to understand any differences in their function, right? Basically, we can say that they actually do the same thing. They both do the exact same thing, and that is they are going to function in cell-to-cell recognition, both intercellular and intracellular recognition. Function in cellular and intracellular recognition. And basically, the layman's terms of this is that these are going to be like name tags and address labels. Function as cellular name tags and address labels. So, for instance, all the cells in my body, they've got bunches of glycoproteins and glycolipids on them, and they say all kinds of stuff. But in the most general sense, all of my cells say I belong to Mr. Tate on them, right? They've got a name tag that says Mr. Tate's cell, right? And then they also have a name tag on them that says specifically what organ or what part of my body they belong to. Okay, so let's say I have a kidney cell, and that kidney cell, for whatever reason, shears off and goes into my bloodstream and then deposits itself in my heart. Okay, I have immune cells that are constantly going around and saying, okay, show me your name tag. And you know, this kidney cell that's in my heart says, here's my Mr. Tate name tag, and he looks at it and goes, all right, that's fine, you're cool. Okay? He says, wait. Show me your heart name tag. And he pulls out his little heart name tag, but it says kidney instead. And he says, whoa. That's a kidney name tag. You're not supposed to be here. And so then he kills it, right? He kills that cell. Actually, more accurately, he tells that cell to kill itself. He says, oh, you're not supposed to be here. Go ahead and kill yourself, right? And then the cell obeys, and that everything works out fine. And that happens all the time where there are cells in other parts of the body that they're not supposed to be in, right? And it's not a problem because our immune system's really good at dealing with it, okay? It only becomes a problem when those cells start to do things like not showing their name tags on purpose, right? So let's say it's a cancerous cell, and it's got its name tags modified. So instead of showing a cell that says, or showing a name tag that says, oh, I'm a kidney cell, or I'm a liver cell, or a heart cell, or whatever, it just doesn't show that one. Okay. So then the immune system cell comes along and says, all right, show me your Mr. Tate cell. He says, or Mr. Tate name tag. He says, it's right here. I'm from Mr. Tate's body. Okay. Show me your heart one, and he's like, misplaced it. Sorry. Right? And your immune system doesn't know what to do with that. It's like, what are you doing here? He says, well, I'm dividing. And he says, oh, well, okay, just this once. And next time, make sure you have your name tag, right? And I mean, it sounds ridiculous, but that's actually what happens. Your immune system only knows what to do, or that specific type of immune cell only knows what to do if the name tags are there. It takes a whole other immune system cell to go in and find the ones that don't have name tags, and they're fewer in numbers than the other ones. Okay, so let's say I want like a transplant. Let's say I need a kidney transplant or something like that. Okay, there's a donor. Someone has a healthy kidney and they no longer need it because they, I don't know, are dead or something. Okay, so I'm like, oh, I need this kidney. Great. And they put that kidney in my body. My immune system immediately says, nope, we don't want that because it says it belongs to somebody else, right? And so you got to find a match, right? You might have heard this like donors and matches and stuff like that. Well, there's never going to be somebody who's 100% match. That's like virtually impossible. Sometimes identical twins are 100% matches, but even then, it's not 100%. It's like 99.9%. So what do we do then? Well, let's say that I find somebody who's a match, right? And we'll take this to a ridiculous level. My name is Mr. Tate, right? It's spelled T-A-I-T, but that's not the normal way to spell Mr. Tate. You normally spell it T-A-T-E, like Famous Tates and Tate's Pizza and everything like that, right? Okay, so anyways, let's say I find a kidney that says Mr. Tate on the name tags, but it's spelled the other way. Okay, well, my immune system's pretty smart, and so I get this kidney, and it goes around, and it's like, hey, show me your Mr. Tate name tag, and it's like, hey, here's my Mr. Tate name tag, and my immune system goes, nope, that's spelled incorrectly, so that I'm going to have to kill you, I'm sorry. And so what they have to do is they put you on these immunosuppressants, and basically what an immunosuppressant does is it makes your immune system dumber. Right? And so the immune system goes around and says, all right, show me your name tag, and it shows him the T-A-T-E name tag, and he goes, okay. I think that says what that's supposed to say. But the problem with that is that if you're suppressing your immune system, you're suppressing your whole immune system, and so you get sick more often and things like that, right? So it's not ideal. But that's basically what you have to have if you have an organ transplant. Is you have to constantly be on immunosuppressants, and when you are no longer on those immunosuppressants, then your body will reject it, even if you haven't been rejecting it for years and years and years. Yeah.
You just answered it.
Okay. Yeah, no, it never gets better. But there is new technology, new discoveries and treatments that are being done using something called xenotransplantation. And xenotransplantation is pretty neat. It's where you take organs from other organisms, like specifically pigs. We're trying to get a lot of organs from, and use those as your transplant organ. So you would get an organ from a pig. Now, they can't do the whole organ so far, but the most common one is a pig heart valve, right? So they can take the heart valve from the pig and cut it out, and it has name tags all over it that say, I'm a pig. So what they do is before they transplant that heart valve in, they dip it in enzymes that eat away all the glycoproteins and glycolipids from the outside, so it just has no name tags at all. Right? Now, if you did that to an entire heart, right, if you had a whole heart with no name tags on it, first of all, it wouldn't work because those name tags are required for a lot of other things, right? But second of all, the immune system would not let that fly. Your immune system would be like, well, that heart has no name tags at all, so we're just going to destroy it. But now, since this is just this little piece, this little valve, right, it goes into your heart, and your immune system might come around and be like, well, this little piece over here doesn't have any name tags. Make sure you add some name tags to it. And so your heart will then add name tags to that little portion that says it belongs to you, and at that point, then you don't have to be on the immunosuppressants anymore because now your heart has put its own name tags on that part. Yeah.
Did you do that with like a liver?
Livers are an interesting case because as they regenerate, as they regrow, so you don't get your whole liver transplanted. You get like a lobe of the liver transplanted, and then it can regenerate, and the new regenerated parts are likely to have the correct name tags on them. So yes, you get less and less likely to reject a liver as it's been in your body, but not like a kidney or a heart or anything like that. And that has a lot to do with the fact that livers themselves are constantly being destroyed and then regenerated by your body. Just in their daily use, they get hurt by processing all the waste and toxins and stuff like that. All right, so That's intercellular. There's also intracellular, right? So intracellular means that if you have a vesicle that's budding off of the rough ER and it needs to go to like the Golgi apparatus or something like that, it's going to have a name tag on it that says, bring this to the Golgi apparatus, or an address label, right? Or it's going to have a name tag that says, go straight to the plasma membrane. And that's how it differentiates between where those vesicles should end up within the cell. Make sense? Okay.